everyone, this is Yazhini. Actually, I have a surprise for everyone, including Snehal and Dr. Karishma.
Yes.
So today I'm going to conduct an exam because I have been because surprise test. So actually, I have been following TTAS for quite some while, and then I realized that these young minds are extremely knowledgeable and they could understand complex concepts seamlessly. This is amazing. And the topic I am going to talk about, it's basically they already know. So it's in fact they are in the teacher level. So I want to just have a test for me to ask. I request young minds to check whether my understanding is correct or not.
Sounds good. Sounds good, Yazhini. So they can quickly type it in the chat window if they have any doubts.
Yes, you can point me, hey, you are wrong. So you can criticize what I
window is already saying no exam, please.
It's like a wait, young minds, it's for me. So you are sort of in the opposite side, unusually. So
sure. Yazhini, have you moved to the next slide? Because we are seeing only the pigment slide.
Yes.
Correct.
Yes.
Yes, we can see it now.
So it's just quite slow. So I am just being kind myself to make it first a thing simple. So what are you seeing on the screen? There are 3 pictures, isn't it? So in the left top, you see there is a building which is made up of bricks. And when you take individual brick and crush it, you will see that there are sands and soils. And similarly, in the right top, you see that chalk, colorful chalk. And then when you break it, you see powder. And if you take in tiny molecule, you won't even be able to see it. And the bottom left, what you see, it's basically Google Map everybody uses. And in the satellite view, you see that all greens and sometimes it's like white, which is filled with buildings when you zoom in. So when you zoom in, what you see, there are buildings, isn't it? So what you can understand from in each pair of figures, you see that from left to right, there is a visible object. When you look down and zoom in further, you see that there are molecules that you cannot even able to see. So so every object made up of basic unit, and generally they are invisible to our eyes. So this is common everywhere. Let's just come into our human body. I'm taking our friend, let's say, call it as a Tina or someone, name it whatever you like. So when you zoom in Tina through microscope, what we can see is organ systems. So the human body made up of different organs, so brain, lung, stomach, and intestine, etc., heart, so on and so forth. And is that the end? When you zoom in further, you see that each organ made up of cells. And so even human body have a hierarchical system. When you take the individual, they are all made up of organs. And when you zoom in further in the organs, you see the cells. Now, you see, I'm showing there are different colorful pictures and they are all different cells and they corresponding to different tissues and organs. Now, you see that we have eyes, hand, mouth, hair, and so on and so forth. And do you think in each of the organs made up of same type of cells? No, right? So that is what shown in the...
Yes, no. You're saying no.
No, right. So that means our body had made up of numerous type of cells, isn't it? And they are all different shapes, different, I'm showing it color for the better visualization. They are different shapes. They are dedicated to perform specific function, isn't it? Now, haven't you not wondered, we are eating the same food and then sort of distributed all over the body, but the cells in different region decided to perform specific function and take specific shapes. So what determines the cells to say you do this function, you take these shapes and so on? So this is sort of interesting question. So for this, outside we see that there are differences visually based on the shapes and function. So to understand that, it is better to look into the cells inside. So
So in the chat window, already there are people who are saying it's the DNA.
Yes, absolutely. So they are actually going to test me. So yeah, to understand, let's look at the cells. So here I'm showing living cells, and there are a number of you are taking the organ and trying to understand the cells. So what do you see in the cell? So this is the individual cell. So you see there is a nucleus, which is here. I hope you see my cursor, and then the endoplasmic reticulum, and there is a mitochondria and cytoplasm, and is covered by plasma membrane, isn't it? And you see the cell is so beautiful and very transparent. You see individual organs very nicely. But is it really the case when you actually look at the cells? And it turns out that your cell has like your cell indeed looks like this. It is so many molecules inside the cell. You have a different organ. This is mitochondria. There is a microtubules, and there are vesicles and Golgi bodies, and this is the nucleus membrane, and so on and so forth. It is so crowded, right? It's like you're going to a market with you can't even walk. You can't even take a single step. It's such a crowded system, but it does whatever job it needed to do. So there are so many molecules, proteins, and so on and so forth. And isn't it look complicated? But it's not really. But when you look deep down, it's like you're taking a brick and then break it down into tiny molecule. What you can basically observe is that the four basic components. One is nucleic acids, proteins, lipids, carbohydrates. I'm sure you all know. Now I'm going to see that the nucleic acids, there are two types. One is DNA and this is RNA. Is it right? This is DNA, right? Is it not? No.
No,
saying
yeah,
no.
they're saying no.
See,
Yeah.
I'm a old person. I was thinking this is this is the DNA. If oh
Kavi
no,
says DNA is double
the DNA
helix.
is a double helix.
Beautiful, guys.
Excellent. So yes, so already you know the presentation. So you are very well. It's like you are basically testing me. So this is DNA because it's a double helix and RNA. So together we call it as a nucleic acids and proteins. Here there is a Rangoli diagram, which is this is how the complicated when you look into the molecular atomic level, and lipids, it has again chemical molecule and carbohydrates. Now there is a common thing. So lipids and carbohydrates and proteins we eat, but cell has its own function. It has to digest whatever we eat, and it has to synthesize for the cells to function. So basically the nucleic acids and proteins are the synthesizing materials. So cells actually synthesizes nucleic acids and proteins, and carbohydrate, which made up of sugars, different type of sugars, and lipid, which we eat, oils and so on, that could be used to give the energy for the cells to produce these molecules and perform their function. So in order to understand, see, we have different cells and they are in different shapes and perform altogether distinct functions. To understand what determines them to do their own job, it is we need to look these components. It's like you received a gift box, and in order to see what is there, you need to actually open the box and then see, and if it is something operatable, you have to operate it, right? So you need to you have to open it and then see what are the things happening. So the main basic components that we have to look into is basically nucleic acids and proteins and so on, other things. So that's what we are going to do it. Now, I'm sure already Snehal had taken about 1 hour class on DNA, and you see that cells inside the cell nucleus has a DNA. But how the DNA is packed inside the cell? So this is the sort of pictorial representation. Let's say I have a rope, okay? So this is sort of the DNA. You can say in the nucleus, you are taking sort of the tightly packed. So this is how, let's say, this is how the DNA would be inside the nucleus. Now, so this is called chromosome. So in the human, I'm sure you remember that there are some 26 pairs of chromosomes, right? Is it right?
Is she right,
26?
guys? Ha!
26?
No, you're already corrected. Yazhini.
Yada says 46.
Right. So
How
46
many?
chromosomes means how many pairs? 23.
Kavish says 23 pairs.
Excellent. So I'm just taking one of the chromosome, one of the 46 chromosome. I'm taking it. I am trying to unwind it. Okay. I'm just unwinding. So that is actually when you unwind, you see that there's a nucleosome. So here it's like a coin you can see in the screen, right? So that is the DNA is sort of wrapped around by a series of proteins called histones. And when you remove those histones, and what you see is if you unwind, you can see that there are, so you can see no single thread. And when you actually unwind again, you see that very tiny lines, isn't it? So this is what it's there in the cell as well, and the tiny line is what we call it as a DNA, and it is a double helix. And I am sure you know that it is made up of ATGC molecule, and there is a rule for their base pairing, right? So in order to what is our aim here, we need to understand the DNA inside the cell. So how do you study them? You have to take the whole DNA, right? Because it is inside the nucleus, and nucleus is inside the cells. So there are two membranes. There is cytoplasmic membrane and you have a nucleus membrane. So you have to break the membrane, right? So first, for that, you take the tissue and sort of grind it, at least make the organs to tissue and then individual cells, and through various of chemical processes, you will get something called the milky sort of thread you can see, right? This is how the DNA looks. So beautiful, isn't it? So now that you got the DNA in the test tube, right? So do you think you can say something about it? All that you can say is that white, it's thread, very thin, and then it's like a milky. But in order to say what it has, because this is the material basically going to decide what cell has to do, what our human body has to do, and animals, everything, isn't it? So how do we understand what this white material is going to say to the cells, so that the cells does its job? Now, once you got the DNA, what we are going to see in the DNA is to find what are the regions. It's like when you unwind it. Okay, there is a question. Okay, when you unwind it, there are regions which we know that it is useful for making a protein. That is called gene. And there are regions which we don't know what it does. And that's what I put it as a question mark. It's an unknown region. It could be functional or non-functional, we don't know. So these are the information we need to understand. So the region which is called gene and is responsible for producing a protein, which is an action machine for the cell. Now,
Yazhini, we can stop for some questions.
Sure.
Also,
Yes.
Yazhini, there's something coming on the screen. You know,
I
something.
don't know. Yeah.
Please move this window away from. Just
Yeah, I thought.
check what that is. But we'll start looking at some questions. Okay, Pratyush wants to know what are lipids or what are fats?
Okay, fats are another biomolecules. Basically, if you take oil, right, coconut oil, ginger oil, you have a many sesame, other oils, right? So it's basically other biomolecule. It has a, but when you look at the atomic level, you have a head group and there is a long tail, which is basically energy source. When you eat a sort of the oil or something, it is also gives us energy. It produces ATP and it also helps in various biochemical reaction inside the cell. If you don't have an oil.
Kavish asks, what are they made of? So they're basically carbon, hydrogen, oxygen, but they have certain fat rings, sterol rings that makes them a fat. So it has a chemical signature that makes it a fat and not a carbohydrate. Okay, that is a sterol ring. Just like proteins have amino acids and carbohydrates have sugars. Basic building block is sugar, right? Okay.
Sorry, I don't know why this is coming up.
Yeah.
I'm not sure why it is the case. Okay, anyway, go ahead. If it is disturbing, please tell me so that I can.
Here, Pratyush says press play. Can we try and press play? Yeah, maybe this is what's happening. Here, down, Yarni. On the right side
No,
of
no,
the
no.
screen.
That's the.
You know,
That's
that is
the
for
slide.
animation.
Oh,
That's
animation.
for the slide transitions.
Acha,
It's fine. So,
Or share.
Yarini, is there anything from the Zoom window that is open in your screen?
No.
Okay. I thought it just might be something from the Zoom window, like the chat window or something like that that's open.
No.
That might be causing. Okay. Anyways, it's fine. We can go ahead. And we'll.
Yeah.
Yeah.
So you got, okay, actually, okay, the slide is gone. So basically we want to see what is the genes, how many genes, and then the unknown regions, or so on and so forth. So that is what made present in the DNA material. And in human, there are approximately 25,000 genes which produces proteins, and we don't know how many unknown genes. So when you combine everything together, that is called a genome, right? Okay. So that is called genome. So in the genome, we want to see how these ATGCs are arranged in each of the strands. So this is the meaningful arrangement of ATGC, which will give us sort of gene, and that gene will give us mRNA protein, that central dogma that I am sure you are aware of. So basically, when you have to look into this whole genome, that process called genomics. You are studying the genome, so it is called genomics. Basically, genomics means that milky DNA what you have in the hand, you see what is the arrangement of ATGC in the entire length of the DNA. How do you study that? It's not so simple. So, okay, you got the white color DNA material, and you want to sequence. It's sort of looking into the ATGC arrangement, the meaningful sequences. So that is what we are going to see, the genomic sequencing. You are taking the genome and then trying to understand the sequencing of the ATGC. And there are 3 steps which is involved in the genome sequencing. And did you know that when you take the human body DNA and then unwind it, how long it would be? Do you know?
Very long.
Very long.
How long? Let's see. Oh, Kavish says it goes till the moon.
Excellent. Excellent. Great. It's like, yeah, when you take your whole body DNA and then unwind it, and then from Earth to moon, it can go back and forth 6,000 times. See,
Earth to moon 6,000 times. That sounds like a skywalk.
Exactly. See, you have that long material in your body, and it is invisible.
Literally, just the stringy material.
Yes, it is invisible. But if you have to analyze, if you want to sequence, okay, what is the arrangement of ATGC from that long stretch, will you able to do it? What can we do? Is there any question? Should I stop or something?
No, we can go on a little bit here. We can stop in a bit.
So basically, in order to make our job a little bit easier, we can break it. Isn't it? We can simply break and then try to see in this short fragments and then see what is inside. So that is what in the library preparation step, where you first take the lengthy material and then break it by a protein, and that is called endonuclease. And in order to understand where it is coming, and then you need to do a further process. For that, we are adding something called adapters. I will explain it later. Once the adapters are added, which is color, the adapters colored background magenta. So once that magenta color DNA is added to it, your DNA fragment, you can unbind. So you have to have the single strand so that you can read what is ATGC, the sequence. And that adapter that we added in the second step is useful to attach onto the chip, which is basically useful where you can attach your DNA material, the single strand, and you can sequence.
Okay, so guys, what Yazhini is doing is telling you the basic steps to sequence or analyze DNA, right? We can't be walking one by one till we reach the moon. to understand what each part of the DNA
And
is.
6,000 times.
That too, that too. That's quite a workout actually. If you survive it, it's a workout. So what we can do is take the DNA, chop it into pieces, attach some proteins and start reading them. Basically, it's a very simple process. Go ahead,
Yes.
Yarni, go ahead. Yeah, so this is called preparing the DNA library. Okay, yes,
Right. Yes.
yes.
So once you attached the single-stranded DNA onto the chip, what you can do, you can add individual ATGC chemical, which is sort of color coded. So when you just throw onto this in that plate, so wherever there is a complementarity in your template, that DNA, if it has a, so the T will attach, and then for the T you have a specific color, and that you will detect. When it adds, it will illuminate and you can detect it. So depending upon the complementarity, you can see what is the sequence. So this is what called sequencing by synthesis. There are many other methods and this is commonly used, sequencing by synthesis.
Synthesis, okay.
So for different colors, so let's say I am not able to see, but yeah. So T has a red color and A has a cyan color, green. So whenever it finds its complementarity into the DNA that you attached onto this chip, it will give the according colors, and based on the color codes, you will see you can understand the sequencing of a sequence of the fragment that you are sequencing attached onto the chip. And once you get this, now there is a bioinformatics is a huge process where you take those the signals and then map it to the ATGC information and try to assemble. So this is called bioinformatics pipeline, so data analysis, where you are assembling the individual fragments into the sort of reference genome that you already know. So generally, right nowadays we have a genome information already available, and then you are trying to map the fragment that you have sequenced and see what fragments are adjacent to each other. By sticking these fragments, you are able to understand what is the sequence of the genome. So this
Yarni,
is
let's stop a little here. It's a
sure.
little complex, so we'll stop. Let's just get it clear. So once
Right.
they cut it into fragments, you can synthesize these small pieces, right? And then you have to assemble. So just think, guys, the amount of data that will be generated if you're sequencing something that can go to the moon 6,000 times, right? So Yarni, there's a question here. Can we fix the DNA back after we cut it? Can you fix the DNA back?
DNA back, yes. That is why we are actually trying to, we are adding some sort of labels. Okay,
Right.
this comes after that. And we are trying to sequence both directions. Actually, that is called paired end. So the DNA has a pi prime, 3' end, right? So there
I
is a
think,
two ends.
yes, yes. Yani,
There
that
is
may
a
be too complex for them. I think we'll just stick to can we fix them back? The answer is yes. We do
right.
fix them back, right? We can just
Yes,
keep
yes.
it little simple. One
Sure.
thing that he says is, what are these colors, Yarni? Somebody is asking, what are these colors? So these are just for our visual representation, right?
Yeah, these colors basically in the final step or the one
I
last
think
one.
this ATGC colors, you know.
So it is sort of the you are basically adding some chemical which gives the color. So you don't know, right, whether it is A, T, G, C. They are all chemical, which is again invisible. We can't see it. So how do we understand whether it is A, T, G, C? It's like you are adding some colorful agent which will discriminate A to G. It's like all four will have a different color, so that we can say, okay, this is A, this is B, C, D. So that's all.
Okay. Okay. So it's just a artificial
Label.
man-made color coding.
Yes,
Okay.
exactly.
So one thing we have to think about is how much data we are going to generate with all of this, Yarni. Right?
It will be GB if I talk in terms of computer
Computer
memory,
language, GB
computer
level data.
GB level, so many hundreds of GB
Yeah.
data. So it is
Wow.
because you are sequencing such a long stretch, right? So you are breaking it, and you have to not just sequencing once, you are sequencing multiple times. It will
Yes.
be better. I'm just using the available resource in the YouTube for a better understanding. Um, uh, yes. So let's say, as I said, you are making a break You are just fragmenting the DNA and attaching the adapters, which will useful to attach onto the chip that in which you are going to sequence. So this is the called adapters. You are adding into both terminal region, and you want to unbind it so that it can bind onto the chip. Now, so each strand it is there, right? And it is going to bind into the chip, and which has a sort of complementarity region so that it can easily bind to the strand, that fragment you made. And now it is sort of, it's going to synthesize. So you are going to synthesize the another strand, because a single strand, right? You want to generate another strand. Why we are doing so? Because everybody make mistakes, right? So in order to reduce the mistake that the errors that we generate, when you have a multiple copies and do the same thing, when you practice it, it will make it is perfect. The things makes perfect. So similarly, when you have a multiple copies of the same strand and sequence sort of 1,000 times the same fragment, you will get more accurate information. That is what this process does. So it sort of sequences. This is called bridge amplification. Just don't bother it. It's basically synthesize the same DNA multiple copies, make it like a one strand into 1,000 strands, and that will be sequenced.
Sure. Okay.
That chemical. So this is what happening. So this is the ATGC, that order, and depending upon the colors, it will bind, and you will have the signals.
Sounds good. Okay.
Right?
So you can see the A binding to T, then T to A, and so on, right? You can, I think everyone can appreciate the correct basis pairing, right?
And different colors. Based on the colors, you will realize, okay, G is binding. So in the template, we have a C. So this is how the DNA is sequenced. And you are sequencing thousands of times, and you have the each time you have that information, and you align it, make it as a that final the sequence, stretch of sequence. Okay.
Sounds good. We can move on a little bit. So basically, from one strand, you build another strand and then get all this data aligned.
So there's a very nice question from Ala. She asks, is there a machine to do all of this?
Excellent. Yes, it looks complicated, right? So it's been like a several sort of 2 decades of work, and now a machine is available. It is sort of 2 days work, that when you give your sample, your genome sequence is available on the pen drive. So that is how advanced our technology is.
So basically, you just put in DNA, or you put larger copies of DNA, and what you get out is a like TTGGCCCCC TTD whatever, like that, something like that, which you have to make sense of.
Yeah. Yes. Now, now we'll go back to our primary question, that in each cell we have a DNA, and then when you sequence all of them, what we found that it's actually all the cells in the human have the same genome, right? So there is no sort of difference in the genome content of the cell. So at the DNA level, we don't have sort of any differences between cells in the human body, but they are different in terms of their shape and the function. So what else can we see so that we can understand the reason why these cells perform different function? So next, let's move on the other nucleotide type, because we are dealing with the basic components, right? We can understand everything in the basic aspect. So we are looking into the next basic component, which is RNA. So I think last session is about RNA. So I know, I'm sure young minds are already well equipped with, know very well aware about the RNA. So in the central dogma, we know that DNA will give RNA through a protein, and then RNA works as a template to produce protein. Now we know that the DNA is sort of common in all cell types. Then we'll look into the transcript, the RNA transcript. So this process, when you look all the transcripts present in the cell, it is called transcriptomics, RNA transcriptomics. Should I stop for some question or?
I think we can go a little ahead and then we'll stop in the next slide.
Okay, so this is another level. So we looked into genomics, and the next level is transcript study of the whole RNA transcript in the cell or organisms, and it is called transcriptomics. Next molecule is the proteins. I think so many talks about proteins, and because it's an action machines in the cell, it is very, very important. So when you look at the whole proteins which is there in the cell, so what is it called? So proteins. Anybody, any guesses from protein? And when you see the whole proteins in the cell, what could be called?
So like
Okay,
gene
so
is
all of
called,
you, we've studied genes,
yes,
and study of all genes is genomics.
yes.
So if we study proteins, what is the study of all proteins called?
What do you
Can
think,
anybody
guys?
guess the word?
Proteinomics. That's pretty close.
That's
Very
very
good.
Really,
close.
very good, very good. Proteinics.
Very close. Protomics. Okay, I see.
Yes.
Everybody's inventing words today. I love this.
Yes, yeah. Proteinology.
Yeah, why not?
Okay, that
Anushka
sounds
says
great.
proteomics.
Someone actually typed something which is Anushka. Anushka. Yes. Yes. Actually, it is called proteomics. So the entire proteins in the cell is called proteome, and then the study of the proteome is called proteomics. Excellent. It's very amazing, kids. Now, my work, one of the work in the PhD is to use the bioinformatics approaches and then study the proteomes of animals, because this is sort of, we are taking the animals and then see what is the differences. Here there are 6 animals we studied, and they are all grouped under called Aprotheria. Just never mind, but we are trying to understand what is the common and the differences in the proteins in each of the animals. And what we observe that in elephant, we see that there is a protein called MAGE-B2. It is very high compared to other animals. Let's say I'll take the Tina. Compared to Tina, the human, elephant has very high number of MAGE-B2 protein. And when we look into the knowledge of this protein and already people what they know, we see that it's also it helps in the successful birth, because we know that elephant sort of spends 18 to 22 months, nearly sort of 2 years to give a baby. Now, if it spends so much time for that, it requires very rigorous mechanism, right, to have the successful birth. So the MAGE-B2 is sort of having higher number could probably help the increase the successful birth. And the second interesting observation what we see that olfactory receptors, it is also a protein which helps in the sense of smell. So we know that elephant has an exceptionally high sense of smell, right? How many know that it can distinguish very minute difference in the smell, which we can't. It's
No,
like
I didn't know that. I always associated elephants with having this amazing memory. They say
memory,
elephants
but
can
the
remember
memory is sort of the olfaction. It's basically through olfaction.
through the long nose, yeah, long
Yes.
trunk, yeah.
Yes, and
Makes sense.
it is basically, yeah, the olfactory receptor is the reason, and the number of olfactory receptors in the elephant is way, way higher than what we have. We are sort of highly evolved for the visual cues.
Yes.
But these animals, all the Aprotheria, that the animals that we have studied is mainly well developed for smells.
What is
And
Aprotheria? I didn't get that, Yazhini. What is Aprotheria?
actually, it's like we call Homo, right? All the human, the taxonomic
Homo sapiens.
classification.
Yeah.
So Aprotheria is like animals which stay in the Africa or sort of evolved from the Africa. So all these 6
So can
animals.
we name some of them, guys? Can you name them? I can recognize the elephant. Can you recognize any others?
It is highly uncommon. Yeah,
Anteater. Then one of
very good. Yes. Yeah.
Yes, there's an anteater. Then
True.
there is a
Mouse. There is a
shrew.
Walrus.
What are the others? Anteater, shrew. One of them looks like shrew. That's very well done. Beluga. What is a beluga whale? I don't know if I see a whale there. I
Oh.
see something like a wolf, the
Yeah.
type one, yeah, that's going ooh like that. And then uh, I don't get two. Is this like a, I don't get the sea creature, maybe a giant turtle or something, or a seal. Correct, it may be a
A
seal.
seal.
Yeah, okay. So these are the Astrotheria. Okay.
Yes, together.
Yeah,
Yes.
yes. Go ahead, Yazhini.
Yeah, the last one like a shoe. It is a shoe in which we found that the ribosomal proteins, which basically helps the protein synthesis from mRNA to protein. This is the protein helps to do that, and we see that it is that animal has a very high number of ribosomal proteins compared to humans, and one sort of fact that this animal always starves. Most of the time, it starves. So because it doesn't find food. In that condition, you need to survive, right? And we find that these high abundance of ribosomal proteins could be involved in survival during starving condition.
Oh, I
See,
see.
yeah. So basically, the study of these biomolecules, we could understand how the animals evolved to adapt their based on their habitat and then food availability and so on and so forth. So this is one advantage studying the basic biomolecular details. So this is at the proteome level. There is a one more level called metabolites. Whatever we eat, small molecules, and then the sugar gets decomposed into other molecules, right? So that concentrations in the cells and their interactions together called metabolites. And when you study all the metabolites in the cell, any guess again? Should I ask metabolites?
Yes, Snehal has already asked.
Okay,
What is the
let's see
study?
what everybody comes up with. Metabology, metabolics, metabolomics.
Yes.
Really? Really? Cool.
Metabolism.
Yeah. Because of metabolism, we get all the metabolites. True. Yes. Great. So it's called metabolomics. Again, Anushka is right. Great. So we can study different levels, the biomolecules, taking different biomolecules available in the cell and see whether those information can help us to discriminate the cells and explain why the cells behave in certain manner, because our DNA material is the same in among different cell types, right? But what we from the metabolomics, proteomics, and transcriptomics, we see that there are differences observed, and those could explain the cell's function and their shapes. So that's what we have. Now, what is the use of knowing this information? One, I had already shown through my PhD project, where we could see the understand the evolution, adaptive evolution of the each animals. But specific to human, if you stick to that, we see that it could have numerous applications. But I'm just listing a few of them. One is personalized medicine. So have you ever noticed if two-person taking the same medicine, one may actually respond, another may not respond? And generally human genomes are same, and probably little bit differences. If you find the little bit differences, hardly only one out of millions of letters, if there is a one letter changes, you can have that drastic difference in the response to your medicine. So when you know this information, the DNA, the genome information, only then we could understand what is the difference on the DNA level. Based on that, which here I have noticed the SNP is a single nucleotide polymorphism. So basically, there is a letter change. If that letter change we could if you could identify, and then based on the differences, we could personally treat individual human being. So that is one advantage of studying the genome. And coming to the cell level, we have a different types of numerous cell types, and study of these different omics, proteomics, transcriptomics, genomics, metabolomics, we could actually characterize individual cell types, and we can understand the biology of the system very well, and how cell differentiates and get its own cell nature. And disease diagnosis. So depending upon the any abnormalities in the cell. Let's say in one cell we don't have a sufficient protein amount, or the protein is not behaving how it should be. So these could be analyzed. That is called disease diagnosis. Only when you study those protein biomolecules and other biomolecules, we could get to know what is the problem. So that is the advantage of knowing this information. And once you get to know the problem, you can specifically target those problematic targets, and then design medicines which could be treated. And finally, you can understand the relationship between animals, which is very useful, and appreciate how the nature is sort of maintain our ecosystem and our living system on the earth. So with that, the main goal of this presentation, not only test myself through this excellent young minds, but also motivate that the cells that we have is highly complex system. It has numerous warriors, tiny warriors, biomolecules, which does right now, it does so much of work inside the body and make us to work fine. So given this wonderful, highly intricate system, we should be feel gratified, and then make those cells happy by being healthy. And in fact, the corona made us to realize that health is very important, and be aware of that. With that, I would like to thank the organizers, talk to a scientist. So without them, this cannot happen. And I see that it has a broad scope to the level that it can actually help the students to decide their career. And the next acknowledgement to whom I should thank. Any young minds, tell me to whom I should thank. Okay, of course, the young minds. Without young minds, how will I present and how this event can go? Isn't it? So yes. So yes, I really thank for all the young minds to listen patiently and sort of testing my understanding. And I would like to thank my PhD guide, Professor N. Srinivasan, and Dr. Sandhya Sagaran, the one of the project which I explained, I did in collaboration with Dr. Sandhya Sagaran, and Shogini, who is an audience here. Without her, I wouldn't have known this event. So I would like to thank her, and school teachers. They sort of like ignition. So they sort of ignited our thirst to learn more and pursue science, PhD, and so on. So I would like to thank, and parents. Without them, this wouldn't have happened where I am today. And thank you for other audience who are in right now listening.
Yeah. Thank you so much. Thank you, Yazhini. Yazhini, we have some questions, and it actually starts with a personal question to you. What is the meaning of Yazhini?
Okay. Okay. So basically, there is in old days, there is a musical instrument called yal. The music comes out of that. It's so sweet. It's like you can forget everything. You just focus on that and enjoy it.
So it's a Tamil word?
Yes, it is a Tamil word. So actually, the Yazhini means the name is sweeter than the music. So that is what is.
Sweeter than the music. Yeah. Yes. So beautiful name. Beautiful name.
Thank you.
Really beautiful name. Very unique name.
Yes. Yeah.
Sohini says she's the TTS ambassador in IISc. Yes. Thank you, Sohini. Thank you. So we have planted our allies. Yes.